Okay, great. I think we can get started, and as people join, feel free to, ask questions, put in the chat if you're a few minutes late. But my name is Carrie Johnson. I'm the Director of Community Engagement at CureDuchenne. Thank you so much for joining our webinar today. I have Dr. Paolo Bettica from Italfarmaco, and he is going to give us an update on the development of Givinostat in Duchenne. So I know a lot of people have been looking forward to news from Italfarmaco, and we're really, really glad to have their team today here to talk a little bit about that. We also have a live Q&A at the end of this presentation. So at the bottom of your screen, please do feel free to use the Q&A box throughout so you don't forget your question. And towards the end, we're going to have Mike Kelly and people from the Italfarmaco team and our research team to facilitate that Q&A. There's also a chat feature, and you're welcome to email us with questions if something pops into your head tomorrow or the next day at cares@cureduchenne.org. And then they will also share their kind of patient advocacy email and contact as well. So I'll let you take it away, and thank you so much again for being here today.
Thanks, Carrie. Thanks a lot for the invitation for this update on the development of Givinostat. As usual, my disclaimer, I'm a full-time employee of Italfarmaco, that is the manufacturer of Givinostat. Givinostat is currently in development and is not approved for sale in any country around the world. And this presentation is for scientific purposes only. So before I take you through the result of the APD study, let me remind you about the mechanism of action of Givinostat. So Givinostat is an HDAC inhibitor and is not acting, trying to restore the synthesis, the production of dystrophy, but is actually acting on the downstream event. So what happens when dystrophin is actually missing? This cartoon is summarizing what happens when dystrophin is missing in the muscle cells. So you are probably very familiar with what are called the mechanical effects. So dystrophin provides some stability of the muscle to the muscle fibers. And when dystrophin is missing, the stability is not there anymore, and therefore the muscle fibers are actually being, if you want, damaged much more frequently. What is possibly less known is that the lack of dystrophin is also impacting on the repairing system. So when the muscle fibers are being damaged, as is the case in Duchenne, then the repairing system does not work properly, and therefore the muscle fibers are being substituted. So when we add Givinostat to the picture, as I said, we are not going to replace dystrophin, but we are actually regulating and improving the downstream effects that I was describing before. So there is a reduction in inflammation, there is a reduction in fibrosis, and an increase in new muscle fibers generation. So if I put everything together, as you can see at the top of the slide, you see the situation in the Duchenne muscle with increased tissue damage, with the death of muscle fibers, chronic inflammation, fatty replacement, and reduced muscle fiber regeneration. So when we add Givinostat, we reduce the tissue damage, we reduce the death of the myofibrils, we reduce inflammation and fatty replacement and fibrosis, and we increase the muscle fiber regeneration. What I've just summarized is actually the result of a number of studies that have been published a few years ago. The first study was actually in the MDX mice. And this was, by the way, also replicated more recently. And then we actually were able to fully replicate these results also in the MD boys in our phase 2 study, where we have shown an increase in muscle fibers diameter, an increase in muscle fibers in general, and a reduction of inflammation, fibrosis, and fatty replacement. So these results were actually the trigger to our phase 3 study, the EPID study, which was a randomized, double-blind, placebo-controlled study that was conducted in North America, Europe, and Israel. It enrolled 179 male ambulant subjects that were randomized 2-to-1 to either Givinostat or placebo. So 2/3 of the boys received Givinostat and 1/3 received placebo. The objective, as you can see here, was to understand whether Givinostat can preserve the muscle mass and delay the disease progression. And the design of the study is summarized here. So after a screening period where also a magnetic resonance test was conducted, the boys were randomized to the two treatment. And let me remind you that Givinostat is actually an oral suspension, so it's taken at home by the kids in the morning and at night. And they were randomized to either Givinostat or placebo, treated for 18 months. And during these 18 months, there were frequent visits to the site, at least every 3 months, to measure the function of the kids, the strength, and obviously also all the safety parameters. And then, as you can see here, at month 12 and month 18, all the boys were also undergoing a magnetic resonance test. At the end of the study, all the boys were invited to take part into the open label extension study, which is still ongoing. Just a brief description of the main selection criteria. So we enrolled boys 6 years and older. They had to be ambulant at study start. We enrolled any type of Duchenne, given the mechanism of action. We really can treat any type of mutation. They had to be on stable steroids for at least 6 months. And then we actually, the functional criteria at entry were geared to identify patients that actually were not expected to lose the ambulation in the 18 months of the study, but that could still have some, if you want, change in the functional test in order to be able to show an effect of our drug. In terms of the endpoints that were measured in the study, the primary endpoint was a mean change after 18 months in the time to climb 4 stairs, obviously comparing Givinostat and placebo. And then we had key secondary endpoints that were related to function as a North Star, the time to rise from the floor, and the 6-minute walk test. They were related to strength as the knee extension and elbow flexion, and were also related to, if you want, an objective assessment of the status of the muscle, which was done by magnetic resonance spectroscopy. So let's move to the result. The study actually met its primary endpoint. As you can see on the left-hand side, you can see the change in time to climb 4 stairs in the 18 months. The blue line is Givinostat and the red line is placebo. And as you can see, the change throughout the 18 months was different in the two groups. So the primary analysis was actually to compare the change in time to climb 4 stairs at 18 months. And as you can see, there was a difference, a significant difference of 1.78 seconds between the two groups. So the Givinostat group has had less decline in the time to climb 4 stairs compared to the placebo group. And we believe that these results are not only statistically significant, as you can see here from the p-value, but also are clinically meaningful if we compare them to the published literature. Then, as I have said before, there were a number of secondary endpoints looking at function, strength, and the MRI. and the secondary endpoints were consistent with the primary endpoint. So if we look at this forest plot that is summarizing the result in the secondary endpoints, you can see that all these blue bars are always on the right-hand side, which means that they favor, the result favors Givinostat. And for parameters like the North Star total score, the cumulative item loss in North Star, and the vastus lateralis MRS, the result actually are less than p 0.05. So let's then look at these results a bit more in details. So this is actually the result of the North Star. And we looked at the North Star in two different ways. The first one is really by using what is called the total score. So you are adding up the score in all the 17 items and you calculate the total score. And then you look at how this total score changes after 18 months. And as you can see, there was a difference of approximately two scores, sorry, at the end of the 18 months between Givinostat and placebo, showing that Givinostat was associated to approximately a reduction in 40% in deterioration of the North Star. The other way to look at it is how many items, so for how many items of the North Star scale, there is a result which is 0. So the boy is unable to actually to perform the item. And you can sum this up, and this is what is called the cumulative item failure. And as you can see, also in this case, we can show that Givinostat actually slowed down the progression of the disease by reducing by approximately 40% the loss of North Star items. A bit more in details, if you're on the item analysis of the North Star, and here you can see the light green is actually Givinostat and the pink bar is placebo. And as you can see, for the items which are most important for the kids that were in our study, so given the age that they had at baseline, there was actually an important reduction in item loss, as I said, for this item which are key for this age of the kids. We also looked at an objective assessment of the Givinostat effect by looking at the fat infiltration in the thigh muscles, applying two different techniques. One is MRS and the other one is Dixon MRI. In the first technique, the MRS, which you can see here on the left, in essence, we are measuring a certain area in the vastus lateralis, which is a thigh muscle that is important for ambulation. And as you can see, the fatty infiltration, which was similar in the two groups at baseline, then the increase in fatty infiltration was actually larger in the placebo group compared to Givinostat. And you can see the p-value here. And the other way to look at it is by applying Dixon MRI. Here you can actually look at more muscles, so not just the vastus lateralis, but also other muscles like the biceps femoris or the whole quadricep and hamstrings. And all these regions are actually key for ambulation. And also here you see exactly the same result of the MRS replicated also by using this technique with highly significant result. So also applying, if you want, an objective assessment of muscle deterioration, the results are consistently showing that Givinostat is actually slowing down the progression of the disease. Obviously, these studies are also very important to look at the safety and tolerability of the drug. And what we have seen in this study is a tolerability profile which was perfectly in line with what we already knew about Givinostat. So the majority of the adverse events were mild to moderate in severity. As you can see, there were only 3 boys that actually had to withdraw from the study because of an adverse event. And then the what are called the very common drug-related adverse event were diarrhea, were reduction in platelets, and increase in triglyceride. As I said, these were actually signals that we had already known about Givinostat when we started the study. So going to the conclusion, the EPID study, as I said, met its primary endpoint, and with the consistent secondary and exploratory endpoints, all supporting the conclusion that Givinostat is actually slowing down the disease, the Duchenne disease. And also in terms of tolerability profile, the study actually showed a good tolerability profile for Givinostat, and the drug-related adverse event, which I summarized before, can actually be managed by appropriate monitoring and by dose adjustment. To conclude, let me summarize what we have learned about Givinostat in these few years. So some time ago, as I said, we started by looking at the effect of the drug on the muscle biopsies. And we were able to show that actually Givinostat was able to prevent the deterioration of the muscle using, as I said, muscle biopsies and measuring by histology. With the result that I have just presented, we actually replicated the, if you want, benefit in terms of muscle morphology using MRI. So looking at not just a tiny piece of muscle, but looking at the whole thigh muscles. And then we were also able to show that this actually translated into a benefit in terms of function and strength. And let me end up by thanking, obviously, all the kids and their families that took part in our studies and are still involved in our open label extension study. All the organizations that have worked, that were very supportive to our development and have helped us quite a lot in designing and conducting the study. And finally, let me thank the clinical team that actually did a very good job in conducting and completing the study. And please note the email address where you can send, where you can ask for further information. And with that, I'll stop and be happy to answer any question.
Thank you so much. Yes, we'd be happy to take questions. At the bottom of your screen, there's a Q&A box, and you can type in there whatever you're curious about. I know we have already a few questions, so I will let Anna, and we'll have Mike Kelly from the research team also here to take it away.
Well, oh, there you are, Mike.
Hey guys. Okay, Paolo, can you hear me?
Yeah, I can.
Awesome, thank you for that. I really appreciate it. Can I kick off by asking about preventative studies in younger children, rather than those that are slightly older and predicted to be in a decline phase?
So let me make give you a couple of answers here. So as you, the EPID study was designed to, if you want, treat kids that were already at a stage of the disease, probably the famous plateau, after which they start declining. But we wanted to avoid a rapid phase of decline, which has been, if you want, the attempt in most of the phase 3 study that actually were not successful, because we were concerned that by, if you're involving kids that were already in a rapid decline phase, we would then have problems with boys losing ambulation and therefore having serious problem in measuring the changes in function. So this is, if you're on a practical problem in conducting a clinical trial. So we actually did two things. One, we selected the boys that were not expected to lose the function in the 18 months, but that were still expected to have some decline. And the second thing that we did was instead of running a 12-month study, we actually conducted an 18-month study. So that allowed us actually to show the signal of Givinostat against placebo. One other thing that is very important, obviously, this is if you're on the key pivotal study for registration. We are well aware that obviously there are some kids that have not been treated with Givinostat, like the kids younger than 6 or the non-ambulant patients. And this is actually something that will happen in the future because as part of our pediatric investigational plan, which is mandatory in Europe, we will be conducting these 2 studies.
Excellent, thank you. One other one before I hand it over to Anna for a few questions. The current formulation was one that was a suspension. Is that the planned commercial product for Givinostat? Yeah.
This is the planned commercial product. So it's a suspension. It's in essence, it is in a, I believe, 140 ml bottle with a syringe. And so the kids take it home. And then, as I said, in the morning and at night, they take their daily dose.
Excellent. Thank you. Anna.
Yeah, you know, I think, Paolo, the point about choosing the kids at the right time to be able to measure it is so tricky because it seems like you need to be sure to find folks who both can do the tests. So someone had asked a question about a 4-year-old, and one of the hard pieces of a 4-year-old is that they might still be improving. They're because of their age at the time in that diagnosis, they might still be improving just because they're 4, not because their Givinostat's really doing anything. And so that's a very tricky thing, but it's something that can be dealt with in other study designs. Paolo, do you want to speak about sort of the open label study and how that plays into answering these really tricky questions in groups that are hard to measure in other ways?
So as we have been conducting this open label study now for, I would say, 8 years, because the kids that were in the phase 2 study also were allowed to actually transition to what is called study 51, which is the open label extension study. So that is, if you're on a study where all the kids that had been treated previously in clinical trials with Givinostat can go to and be treated until the Givinostat is hopefully approved and available on the market. And obviously that allows us to, if you want to have a very important assessment, which is to evaluate when the disease milestone occur. So, for example, loss of ambulation is one, or inability to rise from the floor. And obviously, we can understand which is the mean age at which this milestone occur with Givinostat, and we can actually compare them against the natural history. So obviously, one cannot run a placebo arm in this type of studies. This is unethical and impossible. But obviously, what we can do is to compare these results against a group of kids coming from the natural history databases that, in essence, were identified based on the inclusion and exclusion criteria of the study. And that obviously will tell us what might be the effect of Givinostat on the disease milestone of Duchenne.
Kind of a more real life comparison is what
Correct.
You're saying. So,
That's correct.
after the super controlled part of the clinical trial is done, being able to follow them for 8 years means you can know some of these questions of what happens in the long term.
Correct.
Okay, thank you for that, Anna. Let's just step back a little bit. You know, clearly the trial looked at boys that were stable on steroids, and everyone has an eye to combination therapies, you know, moving forward. Is there plans or thoughts that the company has with respect to looking at Givinostat treatment on top of possible gene therapy or exon skipping therapies, or even some of the more experimental therapies that are out there? What's your thoughts about that, Paolo?
So the first thought is that obviously, given the mechanism of action of Givinostat and the fact that it's acting, if you're on downstream of the lack of dystrophy, the combination is absolutely a very sensible idea. This is certainly something that we'll have to look at. So I would say that at the moment, Italfarmaco is obviously concentrated in making sure that we can submit the best dossier that we can, and obviously in talking with the regulatory authorities to hopefully get a green light. But we are also at the same time looking at all the next round of activities, and certainly trying to understand what might be the situation in a combination is one of the objective of the company.
Yeah, great answer. Like, one of the things I often think about is we have some visibility, I think, to a potential approval for a gene therapy, maybe as early as the middle or late next year. You know, so those products will be out there, and I think being able to understand, you know, what a trial may look like for those types of combinations, along with Givinostat, would be extremely interesting and very informative.
I agree, because obviously we have been talking about the complexity of running a trial, trying to understand the effect of one single intervention. Obviously, running a trial to understand the effect of a combination will probably be even more complicated, but it's certainly something that we will have to look at.
Yeah. Yeah. One other thing, before I hand it back to Anna for a second, is we talked about this online before this started, you know, and trying to get a sense of what is the strategy moving forward with respect to timing and the agency and the FDA, and particularly the EMA. And as you rightly expected, there was a ton of questions, you know, from the audience, trying to get a sense of what is the company's thoughts on this. And I know that you can't be granular, but I wouldn't be doing my job if I didn't ask you for in front of the audience here asking the questions. What is the expectation? What is the timing? You know, from where's your guidance essentially for the Duchenne community?
So I can actually not speculate on the timelines, but what I can say is that be reassured that obviously we are working very, very hard to put together the dossier that will be submitted to the regulatory authorities. The submission of the dossier requires a proper interaction with the regulatory authorities. So if you're on the timing of this is not fully and only dependent on us, it's also part of the discussion with the regulatory authorities. And obviously the aim is to bring this drug hopefully to the market as soon as possible and to as many people as possible, because that is also the other important point. So obviously this is not exactly the timing that you wanted to hear, but please stay tuned, and obviously hopefully this is going to be a rapidly evolving situation.
Yeah, I will say that it's not just you on the spot. This is the same for every company. The questions are always being asked. And I think the audience recognizes one can't be clear. You know, it's difficult to predict when the data set is going to be in front of the agency and how long they will take to go through these things. So, but I do appreciate the answer. It really helps, you know, the Duchenne community really think about this in a different way.
Excellent, thank you.
Yeah, and this is my place to do a plug for groups like CureDuchenne, because people, so if you email the patient advocacy at Italfarmaco group, it comes to me and I'm the one who sends you your answers. And what I will always tell you is that, yes, Italfarmaco is working really hard to use all of the data that the community collected and to get it in front of regulators as soon as possible. But I think it's important for the community to know that there are a lot of other clinical trials out there that may be recruiting now or sooner and might be available, and none of us would want you to wait for an Italfarmaco product if you have something else you could do now. And the best thing you can do is reach out to Mike and the CureDuchenne team, because they can help you find some options that might have concrete timelines that you can plan for now. So that's my pitch to the community. Please, we are so grateful for all the work everyone has done to make the trial a success. And we know that time is precious. So please make use of your resources at CureDuchenne.
Okay, I'll ask one more. Steve, the data's great. Congratulations on it. I've said that to you a few times over the last few months as we've met and talked about it. One of the things that's intriguing to me is the open label extension study is continuing. I don't know how far you're into that, but there's always questions about the durability of the response, or is there an enhanced response as we treat these boys longer? When can we expect to get some insight from that OLE study and looking at what you've actually got and how robust this signal is?
So you might recall that in my previous presentation, we have been showing the effect on the loss of ambulation in the 20 boys that were enrolled in the phase 2 study. And there, obviously, we did not have a proper comparison, but we actually saw a delay in the loss of ambulation when compared or contrasted, if you want, to the published result from the Synergy group. So now we are actually conducting the analysis using, properly matched group. And hopefully in, let's say, few months, we should be able to actually share the results. So also here, be patient, but we'll soon actually come up with some clear information on that part.
Thank you. Anna?
I mean, someone had asked a kind of a couple of questions that I think are tied together of how long, what do you mean by slowing? What progression, like how long of a change are we talking about in kind of real world things? And also the other one of why do you think there's some abilities on the North Star are changing and some aren't, or if we know that yet? Any thoughts on that, on sort of what the data means in real world
so
language?
let me start from the second one. So regarding the secondary endpoints, I think it's important to understand a couple of technicality of a study, because obviously the ability to show on the famous p-value less than 0.05 really depends on a couple of things. One is how large is the effect, and the second one is how variable is the measurement, because the second one in particular will determine how many, if you're on subject, you need to enroll in the study to show the famous p-value less than 0.05. So this is, if you're on a preamble, to say that our conclusion is that the key secondary endpoints are all showing one thing. So if you take them in the totality, they are showing the benefit of Givinostat. Then obviously there are some differences among the different tests being used. But as I said, that might be specific to the test being used and to the variability of the test and all that. But as I said, I think it's important to look at those results as a totality of effect. And as I said, these are favoring Givinostat. Keep in mind one thing, that there are different tests that are being applied to the clinical trials in Duchenne, but there is not the holy grail test. So there is not the perfect one. So I think it's important to look at the different results that will look at probably different angle of the disease progression. And as I said, to look at the totality of the result. In terms of slowing down the disease progression, this I think is, if you're on the more in-depth analysis of what they exactly mean, will come from the analysis of the whole data set that we have. And so also here, I cannot really be specific now, but I'm pretty sure that once we have all the final result, we can be a bit more specific.
So yeah, I think that's a great explanation that, you know, some of the little differences might be because of the test. It might be because Timmy had a great scale on Tuesday and Jonah had a slightly different scale on Tuesday. And that you might have those little differences, but when you put it all together, that's the key piece. Whether it's the open label data, the open label with the natural history and the randomized trial, it tells one story. But to look at the finer details of test by test, that's where you might need to do more research in the future.
Yeah, let me add one piece, because when you design a clinical trials, the number of subjects that you are enrolling in this trial is designed and counted, if you want, just for the primary endpoint. You probably have heard many times about whether the study was powered enough to show an effect. This is actually a calculation that is being done on the primary endpoint, which is exactly what we have done, which sometimes is similar for the secondary endpoint, some other times is not. And therefore you would be needing larger, if you have a number of patients to reach the famous p-value.
I think the other question in the queue that probably everyone else is also thinking, we already talked about younger kids. What about older kids and non-ambulatory? Can you say a little bit more about the European process and what that means? Because some of the US folks might not be as familiar with what the PIP involves and what that means.
Yeah, so in Europe, when a company, if you want, is submitting a dossier for a new drug, they are actually requested to also provide to the regulatory authorities what is called a pediatric investigational plan. This is obviously done with the specific purpose, which is that any, if you want, drug that is being approved in Europe also is studied in kids to ensure that, if you want, the proper adjustments are done if the drug is needed for a pediatric population. Obviously, DMD is a slightly different, well, it's a different situation, because obviously the disease is in pediatric patients, but the still the European authorities is requiring a pediatric investigational plan. And this is usually a long discussion that a company has with the what is called the pediatric committee of the EMA, and then there is an agreement, if you want, between the company and the PEDCO to provide additional information more related to the pediatric population in the context of the dossier submission or following the dossier submission. And in our case, as I said, we have in the plan a study in kids less than 6 years of age, and then also a study in kids that have lost the ambulation. And also here, stay tuned, and in a few months, you will have more information about these two studies.
Anything else, Anna?
For me, I just see two other questions in the Q&A. I don't know if you guys want to speak about it, or Michael, if you want to share any questions that you hear from the community about drugs like Givinostat.
I've touched on quite a few of them. You know, I think the community is quite excited about the data that we've seen so far. And when it gets to this point in clinical development, the questions tend to come around to what is the approval process and timeline. And the one you just touched upon, I think, was a very common one, the non-ambulant patient population that tends to be quite forgotten at times. You know, so we've touched on each one of those. So I'm quite happy with where we are. There's one final one come in if you want to just sort of reach that out about clinical trials in other parts of Europe?
So that is also something that we are considering at the moment. Obviously, Italfarmaco is a European company, so we are concentrating on Europe and obviously North America. But certainly we are also looking in other regions like the Asian region or the South America. But as I said, this is work in progress, so I cannot really comment on when this will happen.
Okay, thank you. I have no further questions, Anna.
I think the last couple is if we've seen any effect on heart function in the studies. And I know that wasn't a primary endpoint, so I'm not sure what all you can share there. And then the other one is whether the open label study is planned to continue, or what are the next steps for that study?
So with regards to the heart, we did, if you want to assess EKG and echo, echo was once a year, and then EKG was every 3 months. And we did not really see much, but the assessment there was for safety reason. So we really wanted to ensure that there was no, if you want, tolerability problem in terms of the heart. So if you want a specific study that can tell us whether there is a benefit, has not been conducted. Also because the age of the kids in this study was probably too young to really show an effect. Having said that, I think there is one very important paper that was published by a group here in Italy in the mice, where actually there was a demonstration of a benefit of Givinostat on the heart in the MDX mouse. So that is certainly very intriguing, a very intriguing preclinical finding. And obviously, as part of the 200 million different things that we would like to do, there is also one.
Excellent. And any plans for the open label study?
Oh, yeah,
and
sorry, yeah.
What happens in the end with that?
So the open label study is going to run in each country where it is now until the drug is finally approved. So if you want, all the kids that are in the open label study will be treated until hopefully the drug becomes available on the market. And then obviously there will be other means by which the drug can be given to the patients.
Excellent. This has been a great discussion, Mike.
Yeah, it looks like, if we have no more questions, I do see one note about kind of receiving this information in other languages. Anna, could you let the folks on the webinar know about the services you guys provide?
Absolutely. So we do have bilingual staff. If you email patientadvocacy@italfarmaco.com in Spanish, German, French, other languages have slightly longer turnaround time. I apologize. We need to get more staff. But we also do have plans to share updated translated materials in the future. So stay tuned for those. They're just not quite ready yet. Everyone's been very busy getting the analysis finished. So we'll be looking forward to that later this year.
That's great. Thank you so much. So thank you, Dr. Bettica. Thank you, Anna, and the rest of the Italfarmaco team for bringing this information to us. Thanks so much, Mike, for helping answer all of these questions, and to all of our attendees. This was recorded, so if you want to revisit this information or share it with your networks or ask more questions, this will be in the CureDuchenne resource library, and you can reach our team. As Anna noted, we talk to families all the time about all of their options. So we would be happy to chat with you about your personal situation. You can email me, Carrie, just my first name, @cureduchenne.org, and we'll set that up. But thanks so much, everyone. This was a wonderful discussion. I appreciate it.
Thank you. Thanks a lot. Bye.
Thanks, everybody.
Bye-bye.
Have a good day.
Bye-bye.
Bye.